Hello, everyone. My name is Michael EWALD. I'm working in the vegetation group at the Institute for Geoecology at the KIT in Karlsruhe. And here I will talk about the reconstruction of the nitrogen status of vegetation using herbarium samples. First of all, it's important to know that today we have an intake of reactive nitrogen into ecosystems that is about 20 times higher than under natural conditions. And this nitrogen mainly comes from anthropogenic sources like intensive agriculture and the burning of fossil fuels. The nitrogen emitted from the agriculture is in the form of ammonia, which is deposited relatively closely to the emission source as ammonium. And the nitrogen emitted by the burning of fossil fuels is mainly in the form of nitrogen oxides, which is also transported over relatively long distances and then deposited as nitrate mainly. This intake of reactive nitrogen into natural ecosystems can be a serious threat for the diversity and especially in nitrogen-sensitive habitats. And we would like to understand the effects of nitrogen in these ecosystems in a better way. And for that, we would like to take a look into the past to better understand the current situation. And one way to reconstruct historic nitrogen conditions is by comparing traits of historic plant samples and of recent plant samples. And this is what we did. So we sampled leaves at herbarium from herbarium specimens, and we sampled leaves from the same species in the fields, and we measured the leaf dry mass, the leaf area, the leaf nitrogen content, and also the delta 15N value of these leaves. And for all of these traits, we expect a positive relationship between the leaf trait and the nitrogen availability at the site. And we formulated two different research questions. One is whether leaf traits differ between historic and recent samples of characteristic plant species in extensively used habitats. And the second question was whether these differences can be used to reconstruct the historical nitrogen status. And for this purpose, we selected 6 habitats that are potentially nitrogen sensitive. And from these habitats that you can see here, we selected in total 12 characteristic species. 6 of them that you see here are characteristic species for calcareous soil conditions. And the 6 species that you can see here are more characteristic for acidic sandy soils. In total, we sampled leaf traits between 2017 and 2019 at 160 locations in southwest Germany. And additionally, we took samples from the State Museums of Natural History in Karlsruhe and Stuttgart. And these samples were taken from herbarium specimens that dated back to the time period between 1820 and 1960. And from these leaves, we measured a bunch of traits that you can see here below. But as I already mentioned, we only looked at the nitrogen values and the area and the mass of the leaves. This is the study area where we collected our samples. It's located in the southwest of Germany. The green points represent the locations where we sampled the leaves for the today's status, and the green points to represent the sampling locations of the herbarium samples. The statistical analysis was quite straightforward. So we calculated Wilkinson rank tests to look for significant differences between the historic and the recent samples. And we calculated a linear model for the visualization of the results. These are the results. What you see displayed here are the coefficients of a linear model that you see here above. Blue colors indicate significant differences between historic and recent samples, and negative values indicate that the traits were higher, the trait values were higher in the historic samples, and positive values indicate that the trait values were higher in the recent plant samples. And these are the results for nitrogen content in the leaves, one time at the mass basis and one time at the leaf area basis. And we see that here for the majority of the species, we observed higher nitrogen values in the historic sample than in the current samples. Yeah, but this somehow, this situation changes a bit when you look at the leaf mass and the leaf area. Here we, for the majority of the species, observed higher leaf areas and higher leaf masses. If we take a closer look to the results, we can see that our species can be fit into 3 different groups. For the first group of species, we observe higher nitrogen contents per area or mass, or higher leaf biomass or higher leaf area. And if we look at the amount of nitrogen in one leaf, for these species, we observe higher nitrogen amounts per leaf under today's conditions compared to the historic ones, which indicates a better nitrogen supply today. And in this group, there are 3 species that are occurring on very poor acidic sandy soils. In the second group, there are species which are characterized by lower nitrogen contents in the recent samples, but this lower nitrogen content was somehow compensated by a higher leaf biomass or a higher leaf area. And when looking at one leaf, we found no change in the amount of nitrogen per leaf for this species, indicating that the nitrogen conditions for this species did not really change. And yeah, in this group, there are 3 species. There are the 3 species which we sampled in forests. And the third group, there are 2 species for which actually the results indicate that the nitrogen supply has decreased. And for these species, we observed lower nitrogen contents today. And this was not compensated by a higher leaf mass or a higher leaf area. And these are the 2 species that we sampled at the calcareous dry grasslands. Here we see the results for the delta 15N values. And yeah, for the delta 15N values, we assumed that a system which is nitrogen saturated, and there we have more losses of nitrogen, and we assume that this escaping nitrogen is isotopically lighter than the remaining nitrogen in the system, and that's why we expected higher delta 15N values in systems that were more that have a higher nitrogen saturation. And actually, we expected higher delta 15N values in the recent samples, but we observed the opposite. And there are 2 possible explanations for that. First of all, that we have a decrease in the nitrogen saturation today compared to historic conditions. And this may be due to an increase of the CO2 content in the atmosphere. But we rather think that this is a sign for the high influence of the ammonia and ammonium deposition from agriculture, because this has a strong negative delta 15N signature. However, we have some difficulties to really interpret these results, and this is due to the strong variation of the delta 15N values of the sources that the plants use. So For example, we have a strong decrease of delta 15N in the nitrate depositions in the time period between 1800 and 1990. And the delta 15N of ammonium is strongly varying with distance, with the distance from the emission source. So, for example, with the distance to a farm. So somehow these values need to be corrected, but we are at the moment, yeah, we're not at the point that we can present some yeah, a good corrected values. And if somebody has experience with modeling delta 15N values in nitrogen depositions, then I would be happy if you could contact me. In summary, we can say that we found remarkable differences in the traits of leaves from herbarium specimens compared to the leaves that were recently sampled, but we didn't find any general patterns across all the species. Instead, we rather found something like a habitat effect, indicating that the species in different habitats, they reacted differently to the environmental changes that occurred. And regarding the reconstruction of the historical nitrogen status, we can see that looking at leaf nitrogen contents is only meaningful if you also look at other leaf traits, because increased nitrogen can also have an effect on the biomass of a plant species, and in some cases not on the leaf nitrogen content. Yeah, and still the results remain difficult to interpret, because we also have a lot of other potential influencing factors for leaf nitrogen, like, for example, the water and the light availability. And regarding the herbarium samples, we also can have some kind of sampling effect. For the delta 15N values, we have great difficulties to interpret these results without a proper correction, but this correction is not straightforward. That's it. Thank you very much for your attention.